Hi everyone! Okay, so in this video I am going to be talking to you about the formation of tissue fluid. So let's just take a moment to put that into context. So we know that blood is pumped via the left ventricle out through the aorta around the rest of the body. It travels via the arteries at high pressure. That high pressure is generated by the contraction of the cardiac muscle in that left ventricle, and that pressure is maintained by the stretch and recoil of the elastic tissue in the arteries and by the contraction of the smooth muscle in the arteries. So we have got blood travelling at high pressure in the arteries. Okay, so this high pressure is often referred to as high hydrostatic pressure, and that's the term that we're going to be using in this video, high hydrostatic pressure. So I just want to think a little bit more about the stretch and recoil of the elastic tissue, just so that we have a good understanding of what's going on. So the value of the elastic tissue is that it allows the blood pressure in the arteries to basically even out. So if you think about the contraction of the heart, you've got this generation of these pulses of high pressure and then low pressure, but you don't want these huge changes of blood pressure in the arteries. And so when the pulse happens and blood flows into the artery, you have stretch. So you increase the internal cross-sectional area of the lumen of the artery. And so there's a larger space, and so this lowers the pressure back down, and then you have the recoil when the pulse is gone, reducing the size of the cross-sectional area of the lumen, maintaining that higher pressure. So this maintains the high pressure in the artery while preventing it from having these massive fluctuations. It's basically Boyle's law, which, if you remember, just says that an increase in the area, if there are the same number of molecules present, will decrease the pressure, and a decrease in the area will increase the pressure. So blood is travelling through the arteries at high hydrostatic pressure. And we know that the highest hydrostatic pressure will be in the aorta, because that is the blood vessel leading directly out of the left ventricle. So the aorta then distributes blood throughout the body using a network of blood vessels, and essentially it branches off. So just beyond the aortic valve, in the ascending aorta, so in the loop going up, there are little openings from which arise the coronary arteries. So you have these 4 coronary arteries that wrap themselves around the heart and supply blood flow to the heart muscle. So these are really, really important arteries. So whenever you learn about things like coronary heart disease, they're usually referring to problems or blockages within the coronary arteries. And then it splits off into arteries that supply the upper thorax, the arm and the head. This is the right subclavian artery. And then the common carotid artery supplies the head. And then you have the left subclavian artery that supplies the thorax, the left arm and also the head. And then it goes down, which into further blood vessels will start feeding into your organs, like your abdominal aorta that feeds into your stomach and your intestines and your kidneys, and then the iliac arteries that then supply blood to the pelvic region and the legs. So you absolutely don't need to know all those names. The only names that you really need to know in terms of, we talked before about names you need to know, but you also do need to know about the coronary arteries. But basically the aorta then starts to branch off. So it goes from being this one vessel to many smaller vessels. So you go aorta, large arteries, small arteries, and then arterioles. So if we were to think about how pressure changes as the blood moves through the body, we would see that the greatest drop in pressure occurs at the arterioles. So our graph would look a bit like this. So the question is, why do we see that drop? The reason that we see that drop is because there is an increased ratio of surface area to volume. This means that more blood is in contact with more blood vessel surface area, so more blood is touching the endothelium. And as we know, even though the endothelium is smooth to reduce friction, there's still friction, and the more that the blood is touching it, the more friction that there is. So this slows down the velocity of the blood within the arterioles and therefore drops the pressure. So why do we want the blood pressure to drop? This is because the blood is going to go from the arteries into the capillaries. Capillary walls, the endothelial walls of the capillaries are only 1 cell thick. they would be unable to withstand the high pressures in the arteries and would burst. Plus, we want blood to travel slowly through the capillaries because we want to provide time in which exchange can occur, exchange of gases, exchange of nutrients, and exchange of waste products. So if the blood continued to travel at high velocities and high pressure through the capillaries, first of all they would burst because they would be unable to withstand the high pressure because they are one cell thick, their walls, their endothelial walls, and two, there wouldn't be enough time available for exchange, so they wouldn't be able to do their job. So then after the capillaries, the blood then enters the venules and the veins, and the pressure remains low. So let's talk now about the formation of tissue fluid. So take a look here. Let's imagine blood is coming in from the arterial end and leaving at the venule end. So this is a really simplistic diagram. We're just looking at one capillary, but we need to understand that it's a capillary bed. So if we were just going to think about it for a second in terms of what it would look like. Okay, so we've got our arterial coming in and then branching into these capillary beds, which then branch down back into venules and back down into veins. So we're just looking at one of these capillaries. Okay, so we know that the blood is coming in from the arterial, which came in from the artery, and then is going to travel through the capillary and then travel out via the vein. So that's kind of just a bit of context what we're looking at. And the other thing that we need to contextualize is that the capillaries are then surrounded. So if we just imagine that this is a capillary for a second, the capillaries are then surrounded by cells. So we've got cells in the spaces around the capillaries, and these cells are bathed in tissue fluid or interstitial fluid. And if we just have a moment to think about the structure of capillaries, we know that capillaries are made up of endothelial cells, so simple endothelial cells, and the whole thing is just one cell thick. So if we were to draw it out, we would see that we are made up of these single layer of cells. With tiny gaps between them. There's tiny holes between the cells of the capillaries. So exchange can happen across the membrane, but exchange mostly happens through these tiny holes. We have got pores between the cells of the endothelial layer of the capillary. Now, the fancy name for these pores is fenestrations, but you don't really need to worry about that. And we know that then flowing through the capillary, we've got the red blood cells that basically fit in sort of one by one, going through the capillaries. And then we've got the blood plasma. And we know that blood plasma is made up of, water. And then it's also made up of dissolved solutes like sugars and hormones and ions and amino acids and fatty acids and all those things, lots and lots of different dissolved things. And then there are also large plasma proteins. There are small proteins and then large plasma proteins, things like antibodies, things like albumins, things like fibrin or thrombin. So lots of plasma proteins. So when we talk about exchange, we're talking about the cell membranes as the surface for exchange. So we're moving things across the cell membrane and we're exchanging things between the cell and the tissue fluid. That's where the exchange is occurring. So things enter the cells from the tissue fluid, and then things leave the cell and enter the tissue fluid. So as we move as the blood plasma moves from the arteries and arterioles into the capillaries, it's going to aid in the formation of tissue fluid, and that's what we're going to look at next. To really understand the context, we've got the blood with all these things dissolved in it, some large, some small, okay, and that's going to be important later on. So here we are. The blood is coming in from the arterial end and it leaves via the venule end. At the arterial end, the blood pressure or the hydrostatic pressure is higher than it is at the venule end. So we can see here that at the arterial end the blood pressure is 40 millimeters of mercury, and at the venule end the blood pressure is 16 millimeters of mercury. So as you are moving along the blood pressure is dropping. Okay, so the blood pressure or hydrostatic pressure is dropping. So it comes in at a higher hydrostatic pressure, and as it goes along, that hydrostatic pressure drops. So it's the high hydrostatic pressure that squeezes everything out. So it's kind of like somebody squeezing on a sponge. Everything is being forced out of the capillary, forced out of the blood plasma to form tissue fluid. So the water gets forced out under this high pressure. Salts, the dissolved solutes, they all get forced out. And as they get forced out, the blood volume drops because all this stuff is being forced out, and it's being forced out through the pores between the capillaries, and it forms tissue fluid. So the blood plasma is squeezed out of the capillary and forms tissue fluid because of the high hydrostatic pressure at the arterial end. Now, there are two forces acting antagonistically. You've got blood pressure acting outwards from the blood plasma or from the lumen of the capillary into the tissue fluid, and acting in the opposite direction, you've got the osmotic pressure. The osmotic pressure is created by a water potential gradient, and the water potential gradient is in the direction of the tissue fluid to the capillary. Now, why is that? This is because some things in the capillary are too large to leave, like the red blood cells and like the large proteins. So they maintain a continuous water potential all the way along. Can you see how the water potential is the same, minus 25, minus 25 at both ends? So the water potential gradient doesn't change, and the water potential gradient is created and maintained by things that remain dissolved in the blood plasma. So the large proteins and the blood cells. So we have these two antagonistic forces. We have one force pushing outwards, the hydrostatic pressure, and one pushing inwards, the osmotic pressure. The water potential gradient, which is maintained by the large molecules in the blood plasma, which are too large to be squeezed out via the pores or the fenestrations into the tissue fluid. And this generates an inward force. Now, at the arterial end, the blood pressure is higher than the osmotic pressure, and so there is a net outward force. We can see that here. So stuff gets pushed out and forms tissue fluid. There is, of course, a second inward force that isn't very clearly shown on this diagram, although it's sort of inferred. You've got hydrostatic pressure pushing fluids out of the capillary, but you also have the hydrostatic pressure of the tissue fluid. All fluid has pressure, right? So there is hydrostatic pressure of the tissue fluid, which is resisting the hydrostatic pressure, is resisting the outward force from the capillary, but it's very low. So who cares? So there's a very low hydrostatic pressure from the tissue fluid, which is resisting the outward force. In addition, you've got the osmotic pressure, which is also an inward force into the capillaries. So closer to the arterial end, the high hydrostatic pressure outwards from the capillary is greater than the combined osmotic pressure and hydrostatic pressure of the tissue fluid. However, as you travel along and the blood pressure drops, whereas the osmotic pressure remains constant, there is now a net inward force. This is really clearly explained in this diagram. So if you take a look here, this is the pressure on the x-axis, and then on the y-axis you've got, first of all, the hydrostatic pressure. So this one here, it's high at the start, and then as we carry along, it drops, and that's exactly what we're seeing happening. So everything in this space represents a time or an area along the capillary bed where the outward force is greater than the inward force. So stuff is being squeezed out of the blood plasma to form the tissue fluid. So there is a net driving force for fluid to leave the capillary. Can you see how the colloidal osmotic pressure is constant? It doesn't change along the whole capillary bed. Whereas at a certain point, they reach equilibrium. So there's neither an outward force nor an inward force. And then after that point, the blood pressure drops below the osmotic pressure. And after this point, there is a force inwards to force the fluid back into the capillary. This is obviously really important. You want to squeeze everything out into the tissue fluid so that exchange can occur. Everything gets squeezed out, so exchange can occur here across the cell membrane. So we can take in oxygen and we can take in nutrients and we can get rid of wastes like CO2 and like urea and uric acid and other waste products that the cells produce, or maybe just exchange of hormones, for instance. That could be another thing that we're exchanging here. And then at a certain point, let's just say here where they reach equilibrium, and then after that point there is a net inward force. Everything is getting squeezed back in. The blood volume increases, the tissue fluid volume decreases, and then the blood plasma will re-enter the venules and the veins and then head off back to the heart. So the blood is travelling slowly along here to allow exchange. The high hydrostatic pressure outweighs the osmotic pressure for the first part of the capillary bed, squeezing everything out. So there is a net outward force. And then in the second half, there is a net inward force because the blood pressure has dropped, but the osmotic pressure has remained the same and everything goes back in. But not everything goes back in. And let's go back to look at the graph to understand that. So if we just think about this quite simplistically, okay, so first of all, there's a longer area over which we're pushing stuff out in comparison to the area where we're drawing stuff back in, and the pressures generated to push stuff out is higher than the pressure generated to pull stuff back in. That means we are unable to draw back 100% of the tissue fluid to reform the blood plasma. We can only take back approximately, give or take, about 90% of the tissue fluid to form the blood plasma. This is obviously problematic. You can't be like, I'm going to dump 100, I could pick a number at random, but I'm going to dump in 100 mils of tissue fluid here. I'm only going to take back 90 mils, and I'm going to keep doing that because your tissues would swell and swell and swell and swell. You'd get bigger and bigger and bigger, but your blood volume would drop and drop and drop and drop, and you would die. So that's no good. So you need some way of getting back that extra tissue fluid to reform your blood plasma so that your tissues don't swell and your blood volume doesn't drop dangerously low. So how do you do that? That's where your lymphatic system comes in. So just take a look at this diagram here. This diagram is showing you exactly the same as we were just looking at. So the blood's come in, blood's travelling through the capillary bed. At first there's a net outward force creating the tissue fluid for exchange across the cell membranes, and then there's a net inward force to take back in the waste products and for the blood plasma to reform, and then it's going to go to the venules and the veins and so on, right? But like we said, we can take back about 90%. So how do we get back the other 10%? We get it through the lymph. So the lymph has open-ended ducts, just open-ended ducts into which excess tissue fluid can drain. So excess tissue fluid drains into the lymphatic ducts, enters the lymphatic capillaries, and then travels through our lymphatic system until it can rejoin our blood vessels. So let's just take a moment to take another look here to see the interrelationship between the circulatory system and the lymphatic vessels. So we've talked about how within our capillary beds, within our tissues, the tissue fluid formation, when we try and draw that tissue fluid back into our blood, we are unable to draw all of it in. So some of it gets drained into our lymphatic system. And then, so that's exactly what you're seeing down here in the diagram. You're seeing the draining of the excess tissue fluid into the lymphatic vessels, where that excess tissue fluid will then travel through the lymphatic system and will eventually return back into the blood vessel. So let's look at how it actually returns back into the blood vessels, just here near the heart. So let's take a look at this diagram here. So if we imagine, so this is our, you don't need to imagine it, you can see it here. Here's our superior vena cava, and we know our superior vena cava is going to lead down into our right atrium, and the right and the left subclavian vein feed into the superior vena cava. So just up here at the neck, that's where we are. Can you see here how the right lymphatic duct and the left lymphatic duct have an opening into the right and left subclavian vein? This is where that excess tissue fluid drains back into the bloodstream. So, a little bit doesn't end up going back into the bloodstream. It just travels through the lymphatic system, and then it goes all the way up, back to the heart, where it'll drain back in at the subclavian vein. And that's a good thing. So basically, by taking out this 10%, that can travel through all these lovely lymph nodes, which are all jam-packed full of white blood cells. And it's a way of taking a representative sample of your tissue fluid or blood plasma and checking it for any foreign pathogens. So it's a really good early defense system or early warning system for your immune system. So if there were any pathogens in there, this is a really good way for them getting picked up and for your immune system to be triggered. So let's just summarize that process. So tissue fluid is a watery liquid that contains glucose, amino acids, fatty acids, ions, oxygen, hormones, all sorts of things. It bathes all of the cells of the body. So every cell in your body is bathed in tissue fluid. It is the site of exchange. Tissue fluid is formed from blood plasma. It is a common mistake for students to say blood plasma becomes tissue fluid, but they are two distinct things, so just be careful in your written expression of this. Tissue fluid is formed from blood plasma. They are not the same thing. So pumping by the, and let's just focus on this bit a second, pumping by the left ventricle wall of the heart, so the cardiac muscle of the left ventricle, generates high hydrostatic pressure at the arterial end of the capillaries. So this outward pressure is opposed by two other forces. One, the hydrostatic pressure of the tissue fluid outside the capillaries, which resists the outward movement of the liquid. And two, the lower water potential of the blood in comparison to the tissue fluid due to the presence of plasma proteins that causes water to move back into the blood within the capillaries. So we've got these two opposing pressures. Overall, small molecules and water leave the capillaries and enter the tissue fluid. Cells and proteins remain in the capillaries because they are both too large to pass across the membrane of the endothelial cells of the walls of the capillary, and also too large to fit through the pores between the endothelial cells. The loss of fluid and dissolved solutes from the capillaries reduces the hydrostatic pressure inside them. By the time the blood has reached the venous end of the capillary network, its hydrostatic pressure is usually lower than that of the tissue fluid. This, alongside the osmotic pressure, forces tissue fluid back into the capillaries down its pressure gradient. But not all tissue fluid is able to return to the capillaries. Some has to enter the lymphatic system, which eventually will drain its contents back into the bloodstream via two ducts that join the veins which are close to the heart, the subclavian veins. Okay, so that is a lot of information, and I hope that it has been useful to you to start to think about how tissue fluid is formed and then tissue fluid is drained back into the bloodstream. So you will need to think about like the structure. That's really important you understand it. You need to understand where the actual site of exchange is, what materials are being exchanged, what causes the tissue fluid to form in the first place, and then what causes the tissue fluid to return back into the bloodstream. So all those different parts of the process. And hopefully these notes that we've been through here will help you to understand that. All right. Well done for listening. That was a lot. Have a lovely day, and I'll speak to you soon.